Thanks very much, Yudong, and thanks for the invitation from the organizers, and I enjoyed the last few talks, so nice to be here. So I'd like to talk today about our work in neuroimage analysis and autism, as it was introduced, and this is a collaboration between our image analysis machine learning group, Larry Stabe, Nisha Dvornik, myself, Xiaoxiao Li, who's now at Princeton as a postdoc, and Juntang Zhuang, as well as Pam Ventola, who's in our Child Study Center at Yale and is a child psychologist. Basically, our goals of this work are to find biomarkers for characterizing autism and then use them to help guide and predict therapy. So autism is a spectrum of disorders. It's basically characterized by impaired social interactions, sometimes language problems and repetitive behaviors. But there's a variety of comorbidities, including anxiety and mood disorders and hyperactivity and other things, and even sometimes intellectual disability. So it's a spectrum because of these range of symptoms and severities. And its prevalence is high. This quote says 1 in 59 in the United States, but I think it's up to 1 in 45. And it's about 4 times more common in boys than girls. And still, causes remain unknown, and psychologists and therapists tend to treat symptoms. Our initial work at Yale was in collaboration with Bob Schultz, who was at our Child Study Center back in the early 2000s. He's moved on to the University of Pennsylvania. But in our earliest work, we looked at structural magnetic resonance images, trying to look for things like volume changes indicative of autism. But Bob brought us into looking at functional MRI, task-based fMRI. And his earliest studies were to look at, trying to look at the differences in reactions to alternating 24-second faces, houses tasks with subjects in the magnet, with the idea of trying to bring out an emotional response, perhaps, or recognition of faces. And so the initial study was about 14 autism subjects and 14 controls, young male adults. And using just general linear methods and this task-based fMRI, Bob's work found that in the young male adult control, the normal subjects, both 2 areas lit up in response to this task. A blue-purple area that was a parahippocampal place area, really recognizing that objects are present, as well as a fusiform face area showing recognition of the human face. But in the autism subjects, only the parahippocampal place area lit up, implying perhaps that there was no real recognition of human forms or faces, and that the autistic subject just saw faces or houses as simply a thing or a place or an object. So fast forward to about the early 2010s, Bob left. Kevin Pelfrey became our new collaborator at the Child Study Center. He's also since moved on to the University of Virginia, but we still collaborate. And around this time, Archana Venkataraman came to us, graduating from Polina Gollin's lab at MIT as a postdoc. And Archana said, let's relook at this problem in a different way. And particularly, let's apply the neuroanatomical atlas. She chose the Destrio atlas of about 150 regions. And then look at correlations between these regions, as the field had been doing and is doing, to get these connectivity maps based on basically Pearson correlations. And there was one set of data, or one set of subjects for the typically developing control subjects, and another set of data for the autism subjects. And she hypothesized or posed the idea that there were underlying neurotypical and latent organization of autism spectrum templates that could be found and uncovered, guided by the idea that there were communities or groups of regions that might work together in a certain way. Basically, the idea here was to look for connectivity, or she called synchrony, and discretize into low, medium, high, or simply low and high synchrony or high connectivity. and then compare group data between regions to decide if the synchrony was lower or higher in autism or in normal groups. So this was then set up as a joint probability density function with the two data sets, B and B star for connectivity, the underlying F and F bar, the functional organization of the connectivity, and then the region membership in one type of community or another. Given that there were so many communities, just a few were posed in certain prior parameters. So in this work, Archana used expectation maximization to solve a maxima posteriori estimation problem and estimate the underlying synchrony or connectivity maps and whether any given regions belong to one of just two or sometimes three abnormal functional communities. The task was a little more sophisticated than faces and houses, but tried to do the same thing, basically that there was a human light point form of trying to bring out a human response, and then scrambled a pattern of just these lights these tasks called biopoint in alternating 24-second increments. So in this biopoint task, there were 72 children, ASD children, 43 age-matched controls. And here are the results using this Bayesian estimation algorithm. With two communities posed, the pink community that was found was declared hyperactive or hyposynchronous in that there was increased connectivity in autism subjects, F bar being greater than F for the regions in that community, and a blue or hypoactive or hyposynchronous community where there's reduced connectivity in ASD, F bar being less than F. If you took these regions and mapped them out onto this popular neurosynth database that's gleaned out of the lots of papers in the neuroscience literature, you can see that the hyperactive community maps onto things that are strengths in autism, like visual perception and pattern recognition. But the hypoactive community, more prevalent, better connectivity, reduced connectivity in autism, mapped onto these more emotional regions. So these are strengths in the normal subjects, but weaknesses in autism. So all of this is group data. And we wanted to get to individual and subject analysis in order to guide therapy. So the next evolution of our work was to move towards individualized biomarker detection from task-based fMRI again. And we started doing a classification task and then using this task then to uncover biomarkers. We started this work using intensity-based data, using convolutional neural nets and data-driven work and ROI ablation analysis to see which were important. But we most recently evolved to using graph convolutional networks. And this work's shown in MICI 2019 and 2020. So let's look at this setup. Basically, as other groups have certainly done, we think of the brain mappings as a graph. We switched to the Desicon-Kiliani atlas, which posed about 84 regions of interest. And so the graph nodes are each region of interest plotted here as a graph. Hanging on each node are node features, and started with about 100 features in each one of these regions, including the bold signal mean, the bold signal standard deviation, Pearson regular correlation, if you will, to all ROIs, and then the ROI coordinate. The node edge or the graph edges or connections were formed by basically control correlations between regions, not controlling for everything else or all other relationships. Basically a partial correlation, which gave a sparse connectedness for the E or the edges. Now we, unfortunately, as Dave was talking in the last talk there, we are also using small sets of data sets for a variety of reasons. So augmentation was important by basically resampling training data to get resampled values and build up the database. So the processing pipeline for our graph neural net brain network analysis We have task-based fMRI data, brain parcellation using the Duncan-Kiliani atlas. And in each region, there's basically mean time series that come out from that. We do a correlation, in this case, both Pearson and partial correlations, and you can represent it here, and then change that into this functional graph. The graph network processing then reduced it to, after graph convolutional layers, to fully connected layers to do the classification. And from this, we infer communities, do classification, and extract biomarkers. We could use this same scheme for prediction, but we haven't moved that. We do that a different way, excuse me, as you'll see later. You might say, why graph neural nets? Well, we like the scalability of adding features and nodes. As you'll see, they turn out pretty effective. And because of the sparse nature of the nodes, they're quite efficient. And also, I would argue that they're a little bit more directly interpretable in many cases. So here's our brain graph neural net, BrainGNN. Take the initial graph and run it through these graph convolutional blocks, basically 2 of them in series. Each one of these blue blocks has 2 blocks embedded in it. One, a graph convolutional layer, or a node convolutional layer. And this basically is aimed at, where here the node features are changed and are reduced to some efficient information, but the graph structure doesn't change. And then in a second step, there's a node pooling layer where the node features don't change, but there's a reduction in the number of nodes to make it more efficient. And then at the end, everything's aggregated into a vector. So the first stage then is this graph convolutional layer in this classification biomarker and recovery stage. We call it region-aware graph convolution. And we would say that in the traditional graph convolutional net, all nodes are treated identically. Basically, you're applying the same kernel to all nodes. But when the nodes have spatial relationship information, and we're going to assume this and embed it using the community ideas that I talked about earlier in our group work, we want to use this to reduce the number of features at each node, basically based on ROI-aware relationships. So we start with a we have to determine and declare the number of communities, and then declare a random initial community assignment. And then the processing will do a conditional kernel embedding based on the communities and do a feature reduction. Basically, what this means is we're assuming a basis function for each beta for each community, and we get a weighting alpha and an offset B that sort of filters down the overall weightings to map them into this community-based space that'll reduce the features. And furthermore, we start with the H sub I features at each node and the E weighting to the surrounding H sub J information, and get out a set of weights. But this new set of weights are going to be based on this setup in the community representation. And in this, I would just note that alpha I sub IU is the membership likelihood of ROI I in community U. And again, beta sub U is each community's basis function. So we start with the raw features, and then after these sequential steps of our region-aware convolution, get a new H tilde, and that is based on the relational information, the surrounding information, but embedding the idea of communities. These are the weights that have to be learned, both ultimately the basis functions, the offsets, the weightings alpha that then end up weighting the W's. All of this is done to reduce the feature set, as I said, and is done for all the processing. So if we start looking at it, again, using these same 72 ASD and 43 healthy controls, we have to declare the number of communities. We declared 8 for starters. And this came out of work from a sister lab at Yale, Todd Constable's group and Emily Finn, where they looked at community clustering in autism and came up with that number approximately. This is from these data, and you see the 8 communities clustered here. And the community membership, as I said, is declared by, here's all the ROIs, the 84 and the Desikan-Kiliani Atlas. And then here's the 8 different communities listed here. And community membership is declared after looking at the alpha values and usually thresholding for only those that are greater than a half in order to cluster into the 8 communities. So that's the first stage. The second stage of processing is this pooling stage where we do salient ROI selection. And as I said, then from the first stage, we reduced the number of features now. Oh, I wanted to point out, actually I meant to do that back here, that basically you could think of all this as kind of a PCA-like reduction into the principal components that are the basis functions of the communities. So by limiting the number of communities from the maybe many, perhaps 50 or tens at least, if not 100, to just 8 communities, we look to reduce the number of features at each node to about 10 with H bar. And then we do a projection. There's different ways of then doing pooling. You could do magnitude-based reduction. We project onto a virtual pooling vector that's normalized between 0 and 1. And then giving values to all the different nodes and their feature sets. This ultimately yields a node score assignment of different values. And then basically we do a top K-like reduction to just keep the high scoring nodes. So it keeps the same number of features, but reduces the number of nodes. As perhaps some of you know, there's some limitations on top K pooling in terms of the ability to regulate pooling scores, and perhaps some issues with rankings different for different input instances. Because of these limitations, we propose to do pooling using an overall loss function for the training of this overall graph convolutional neural net scheme, and embed these distance and group level consistency losses into the loss that's just the cross entropy training of the training data, the original data. So LCE is classification loss. The second term is a distance loss that enlarges the distance between important and unimportant nodes. And the final term is this group consistency loss. And what this allows is whether we want it to be fully individual terms or learning, or we allow some group training from the data set or consistency towards the group data to come into play. So here's that loss function just repeated, just noting that the group level consistency is basically taking the pooled data and saying each individual subject should be close to it over all the regions based on a norm. So the final term, as I just noted, is to do this node reduction. And I would just point out here's that when we weight the group consistency loss very low, like 0 perhaps, you have more of an individual biomarker. And as you weight lambda 2 higher and higher, there's more common important ROIs over the training data. So you move towards a group biomarker, although probably the answer is a compromise in between the two. And in fact, here's 3 visual results of 3 different subjects looking at the left-hand, right-hand side, and then with 3 subjects here plotted in the columns with lambda 2 equal to 0, 0.1, and 0.5. And as you see in these circled regions in each of the subjects, as you apply or enlarge the lambda 2 values, a little more smoothing in here. Now, we haven't mapped these to the NeuroSynth database yet, but we do feel we're getting plausible regions that are reasonable biomarkers compared to our former group data. So we are, these are some selections out of these 72 actually ASD subjects. The classification testing overall, so remember we go through these early convolutional stages, then do a fully connected reduction to do classification, and that's where the previous biomarkers were extracted from. But just to look at the classification accuracy and compare to other methods, we augmented the data 30 times, the 72 times 30 and 43 times 30, the ASD and control subjects to get these kinds of numbers. So we had over 3,400 data sets, divided that into 5 folds, so it's about 690 graphs each. use 3 folds for training, 1 for validation, 1 for testing, and then do rotate that and do a cross validation. So if we compare the different methods in the literature, some simple methods, and here's the number of parameters you need to learn on the bottom here, times 1,000. So that's just 3,000 parameters, 138,000, et cetera. Support vector machines, the classification accuracy is only 62%, same with random forests, but you do have less features. When you get to multi-level perceptrons, a little bit better, but there are lots of features. A convolutional neural net based on just intensity time series data, you are getting up to pretty good accuracy, but you have over 1 million parameters. And then different versions of our own work and work in the literature got a little better with graph nets. Here's our sort of current approach for the BrainGNN. We're getting up to 78% accuracy, which is some of the best we've seen in comparison. And we have a mid-road, sort of about 41,000 parameters that we need to train in these networks. But we feel the work is promising. We're getting towards individualized biomarkers, and we're encouraged. So now I'd like to switch gears a little bit and talk about prediction. And can we predict outcome from baseline information according to certain ASD severity scores? Now, ultimately, we want to use the classification-based biomarkers to steer or guide attention to different regions. But in this initial work, we used all the regions on another atlas. But where we're headed is the most effective autism therapy seems to be different cognitive therapies. And at Yale, one in particular that's been doing well is pivotal response therapy. And this is our collaborator, Pam Ventola, and her daughter, who doesn't have autism, just sort of showing the therapy. It's a really intense therapy over 4 months with every day 5 or 6 hours of commitment to work with the children to try to teach social skills and bring out social responses. There's some no one size fits all, but it's important that early intervention is done to help these children, these subjects. So can we predict treatment outcome from baseline imaging measures is the question. So we again did task-based fMRI on these children at baseline using a variation of our biopoint task with human like forms as 24 seconds alternated with scrambled light patterns. Here are the acquisition parameters, about 3.5 cubic millimeter voxels and these alternating coherent and scrambled light point patterns. We only had 21 children to start with because these are such difficult 16-week treatments. We're now gathering a larger and larger database, and there were 14 male and 7 female. Typically, the early work was done with subjects or participants who had reasonably high IQs for autism children. And the measures that we had were both pre and post-treatment severity scores, a variety of autism diagnostic observation scale measures, but the one we focused on was the social responsiveness scale score and asked, could we predict that? So the idea is to predict treatment response. We looked at percent change in the social responsiveness score between post and pre-treatment with cognitive therapy. And can we predict this based on pre-treatment baseline scan information only? And it might be helpful to know that the lower the SRS score, the better the social function. And the idea with all of this is that predictive modeling could help guide treatment assessment and encourage parents to participate in these difficult and involved cognitive therapies. So in this work, Nisha Dvornik, my colleague, and worked with us and helped develop this scheme, was published initially in MICI 2018. And here we used an automated anatomical atlas. About 90 regions were used, neuroanatomical regions. Within each region, there was a time series data set that was pulled out. And we included phenotypic information, as you'll see, mainly age, sex, full IQ, and initial SRS scores, and ran it through a learning algorithm to do a regression and to get the change in output. And as you'll see, this learning algorithm we used initially in all the regions was a recurrent neural net based on long short-term memory modules. And then the outputs were this treatment outcome, as we said. So this is an interesting idea. I really learned, I enjoyed, we're still working with these LSTM ideas, long short-term memory recurrent networks. And perhaps, as some of you know, these LSTM modules are each made up of sets of learnable weights, basically 4 layers of these: a forget gate layer, an input gate layer, a current state update, and an output layer with a variety of connections. Again, because of the obviously lower amounts of data, we had to do some augmentation. So we randomly sampled voxels in each region and replaced the time series in those with time series information from the training data randomly. So the 90 AAL regions were used, and we'll steer these later on biomarker analysis, as I said. So we sampled the voxels in the ROI with replacement, compute the average time series, do this data augmentation resampling about 50 times. Here's the architecture for the recurrent net. So there's, we looked at 156 time points, basically seconds, and each time point or time window had its own LSTM cell. And a vector of ROI values, 90 of these, were put into each cell. So for each cell, then, there are 8 LSTM nodes, as I showed earlier, and for each node, there's 4 sets of weights that need to be learned. All this is task-based fMRI, as I noted, and they run sequentially and then go through a fully connected output layer to do the regression or prediction. We added the phenotypic information as an initialization, basically, to initialize the first hidden state of the first cell, and then connect the phenotypic inputs to a dense layer representing the initial hidden state and the initial cell state, and then propagate it all and pass it. So using that architecture and the basically 20 subjects for training times 50 augmented data sets, we got to about 1,000 training subjects, and then 1 test subject was used with a leave one out rotation to do cross-validation. And just again, reminder that 90 regions per time point. So here's the algorithm predicted outcome, and here's the true outcome, which is change in the social responsivity score. So if you just use the raw image data, there's a correlation between predicted and true of just about 0.35, although it is significant. If you add the bootstrapped fMRI information, we get up to about 0.53. And if we add the augmentation of the initialization with these phenotypic variables, we're getting up to a promising 0.77 correlation. And I would point out that if you used any of these variables alone, there's basically no significant correlation at all. In fact, it's really low, below 0.1 or so. So this really adds something to the prediction problem, and we're quite encouraged and are further pursuing this work, now again steering by the biomarkers from classification. In fact, here's our next directions. We want to include the BrainGNN, gives us basically nice spatial analysis, and we want to divide up the time series into windows and do these over all these time windows, and then put it into the retaining LSTM recurrent neural network. And we'll do both classification and prediction, steering perhaps the second stage with biomarkers from classification. And we call this the dynamic spatiotemporal graph. So that's the work. I'd like to summarize by saying we feel these new graph convolutional methods for biomarker identification are promising as early ASD indicators. This machine learning strategy is also promising for predicting response to behavioral therapy, initially using the recurrence strategy. And both in general for this work, the data-driven networks are important for both of these identification and prediction tasks. Complete time series information can be incorporated. The grouping into communities and subnetworks seems promising for interpretation as well as computational reduction, although we did show that comparison to traditional models. And our next directions are there's a lot more resting state data around, so we might do some transfer learning to the task-based data, and also try to look towards using that for looking at invertibility and explainability in the networks. We also have a collaboration with our MR spectroscopy group to look at metabolic biomarkers in addition to fMRI. And personalized and individual analysis is key and will be our future focus. This work is supported by NIH, the National Institute for Neurological Disease and Disorders, through this grant. Thanks a lot for your attention. Thanks for the invitation, and most happy to take questions.
Thanks, Professor. We know how busy you are. We want to thank you for squeezing your time out of your busy schedule and for your insightful and thought-provoking presentation. And we, on behalf of all our members of our market conference committees, and particularly our conference chair, Professor Ray-Dan Su, several in our audience have expressed appreciation for the information you presented, particularly on the brain graph neural network and the ROI-aware graph convolutional layers. And they said that they were unaware of those networks. I will pass over to our audience. Here we receive a comment from Dr. Zhou. He asked, thank you for a very inspiring talk. On the graphs of ASD, can you provide more details on how data augmentation is performed on the graphs?
Sure. So for the prediction graphs, that was this slide, and basically, maybe I went through it fast, I apologize. Basically, it was, we consider each regions randomly, and then the voxels in those regions, and just sample them, sample a certain amount, and then go to the training data, and for the sampled voxels, let's say even up to 50% of them, just replace them with random time series data from the other training data. So basically, it's just the augmentations. You have training data, you have the regions, you look at them randomly, you look at the voxels randomly, and you do a sampling and replacement with training data in order to build it up. And that just gives you new instances and new examples. For the classification, it's basically similar, I forget, for the graph nets, it's also basically the same rough idea. We just go in and, I don't know if I have the right graph here. It's just, again, just sort of sample the nodes, sample the features, and do random replacement. Sorry, I don't remember which slide I put it on. But it's the same idea, just trying to do sort of augmentation and get variation in the data as best as possible.
Oh, okay, thank you. And I received a second question from one of the audience. And so I forwarded the question in our Q&A. The question is, the loss consists of 3 parts. What are the meanings of these 3 parts?
Okay, let me go back to that. Yeah, okay. Oh, all right. So, yeah, so here's the loss. Yeah, thanks. So, right. So the first one is the overall network loss. So that's the cross entropy loss. You have training examples where you have ground truth, let's say in the classification, you know whether it's task, you know whether it's ASD or normal. And then you have the input graph, and that's you're just training up whether the overall loss to whether it classifies accurately or not. These other 2 terms are important for the pooling layer. And I had other slides, but I don't think I have them in here. This one is just basically looking at pooling distributions, and you're trying to sort of force the top K loss to enlarge or emphasize the difference between important and unimportant nodes based on these node scores from the projection onto a pooling vector. And the final one is this group consistency. It's a little harder to understand, but it's basically, I showed it here, it's just an L2 norm for the information in that region from the group training data. And for that individual subject, what's the difference for that region from the group data? And you can weight that, as I was saying, to include a higher or a less weighting as to similarity to the group data. So the three terms are basically the normal cross entropy loss for training, a term to sort of emphasize the differences in the important and unimportant nodes for pooling, and an overall consistency to the training data, which is something you can kind of dial up or down and see. It's really empirical at this point of how to set that lambda.